Biomolecules.
Come a little closer to the mic, yeah, if you can.
Yeah, so biomolecules, bio means life and molecules means group of atoms. It is not only the composition of elements that distinguish a living matter from non-living matter, but it is the relative abundance of certain elements that is higher in living matter than in non-living matter.
Yes.
Then we studied about proteins and biomolecules is divided into classified into proteins, lipids, sugars, nucleic acids. And first we discussed about amino acids. It has amino group and carboxylic group.
What are amino acids?
It is building block of every protein.
Yes, right. Okay, continue.
It has an amino group and a acid group.
What is amino group? NH2. An acid group is the carboxylic acid group. COOH. Yes.
Then acids are divided into, we studied about acids. Acids are divided into inorganic and organic acids. Then let's discuss about the R group. The R group is a variable group in the amino acid, and the nature of R group decides the nature of amino acid.
Yes, right.
Then
and we discussed about different types of amino acids, right? And the zwitterionic form. What was that? What is the zwitterionic form of amino acid?
In a zwitterionic form, both carboxylic acid group and amino acid group are in ionized state carrying opposite charges.
Yes, right.
Then
on the basis of the amino acid group or the amino group or the carboxylic acid group, amino acids are of 3 different types. Yes. What are these?
First one, based on the number of amino or carboxylic group. First one is acidic amino acid. Example is glutamic acid.
Glutamic acid. Yes. Aspartic acid.
Second one is,
Did you go back and find more examples for this? I gave you this as a homework, both of you. Yes, no?
Yes.
Yes. So what are the other examples of acidic amino acid? Aisha.
Yes, sir.
Have you noted it down?
Aspartic acid.
Yes, aspartic acid and Iram?
Histidine.
Histidine.
Yeah, histidine.
Okay, Iram, have you done this?
Yes, sir. I found all the 20, but I don't know which one are acid and which one is basic.
So yeah, but that was the homework, right? To find more examples of acidic and basic amino acids, right? So Acidic is aspartic acid, glutamic acid, and what else? Aisha is saying histidine. Do you think histidine is also acidic?
No, it
No,
is
it's
basic.
basic.
It is basic, right? Yes. Lysine, histidine, arginine are basic. Arginine are basic. And the rest one are neutral. Okay. And on the basis of benzene or cyclic structure, we will study cyclic structure today. There are aromatic and non-aromatic. So this I already told you, there are only 3 aromatic amino acids: tryptophan, tyrosine, and phenylalanine. Okay. And then I ask you to go back and read about aromatic ring structure, find 20 different amino acids, look at their R groups. So I hope you did that, right? You did that? Are you familiar with amino acids, their R groups? Which is the simplest amino acid, if I ask you? Let's start with this question. What can be the simplest amino acid?
Glycine.
Glycine. And why is it simplest? It's very simple. See, if you just understand the logic, every amino acid has a primary carbon, and you know that on one side there will be NH2 for sure. On the other side there will be COOH for sure. And on one side there will be hydrogen for sure. So the rest is R. Okay. So in case of R, what could be the simplest thing you can put here to make the simplest amino acid? What should be here in place of R? What should be here so that it is the simplest amino acid? A hydrogen, right? If hydrogen is there, which is acting as the R group, then this amino acid is called glycine. That's why it is the simplest amino acid. You understand?
Yes, sir.
And after glycine, again, so this is going to be same for all amino acids. If in place of hydrogen, I attach it to one more carbon. The moment I attach it to carbon, there will be 3 hydrogens, right, attached to the carbon, because carbon has to fulfill the valency, right? So it will be CH3. This group is called methyl group. So if R group is equal to hydrogen, then it is glycine. And if R group is equal to a methyl group, I hope you understand why it's called methyl, right? Meth, eth, prop, bute, you know these things?
Yes.
Yeah. So this becomes alanine. Clear? These are the 2 simplest structures of amino acids. Is it clear, both of you, Aisha, Neeram?
Yes.
Yeah. Okay. Now, in today's class, we'll start with the next important biomolecule called lipids. Now, before we begin this, you tell me, where have you studied lipids before? In cell, we talked about lipids, right?
Fluid mosaic.
Tell me.
Fluid mosaic.
Yes. So lipids are the most important component to make cell membrane, correct? Very good. So fluid mosaic model, there you have studied that lipids are basically fluidic. Proteins which are embedded in the lipids can move, right? But they cannot flip flop, but they can move their position. We did a question also on that. Yes? Remember?
Yes.
Very well. Now tell me what else you know about lipids already from cell chapter. You have studied the word lipid somewhere else. One is lipid bilayer, which is the plasma membrane. Apart from that, something about its synthesis. Which organelle is responsible for lipid synthesis? Where is lipid synthesized? Where it is packaged? Remember smooth endoplasmic reticulum and Golgi bodies? Iram and Aisha.
Yes.
Go back to the notes and see, because you're just saying yes, and that yes has no conviction. The yes is so blank in itself that it's even worse than a no. Do you understand what I'm saying? Smooth endoplasmic reticulum synthesizes lipids, whereas rough endoplasmic reticulum has ribosomes attached and synthesizes proteins. Remember this or not? See, even not even a yes is coming now.
Yes, sir.
Okay. Okay. So let's go to some characteristics of lipids. Write down characteristics of lipids. You have lipids all around you. You use lipids. They are made up of lipids. But what are the general characteristics of lipid? What kind of molecules are lipid? What do you think? We use a word called fat, right? Fats. What are fats? Or fatty acids, if you have heard of this word. They are all lipids, different kinds of lipids. Lipids, first thing, they are water insoluble. They do not solubilize in water, right? Yes or no? Lipids are water insoluble. First criteria. Next, what do you know about lipids? What are fatty acids? Lipids can be simple fatty acids. Again, there is an acid term. So the acid which will be present here also is carboxylic carboxylic acid. So fatty acids are nothing but this structure. So there will be a R group. Again, this is a variable group attached to COOH. Okay, this is a fatty acid structure where this is the carboxylic acid group. Okay, and this is a variable group. So if you start with the simplest, this R can be CH3, right? CH3. If that R is CH3, what does the molecule becomes? CH3COOH. Remember that? People, yes or no? At least say something so that I know that you are understanding. So write down a fatty acid. A fatty acid is a carboxylic group. A fatty acid is a carboxylic group attached to an variable R group. Attached to a variable R group. Now this R group can be any number of molecules attached. So it is like a long chain. For example, some examples of fatty acids are palmitic acid. This acid is used in many beauty products as well. So it has a carboxyl group. Sorry, I'm so sorry. It's not a carboxylic acid group. It has a carboxyl group. Write down. COH. It's fine. And this palmitic acid, it is CH3 followed by a long chain of CH2, which is repeated 14 times, and then COOH. This is palmitic acid, and in a palmitic acid, this was asked in one question. You can write down palmitic acid has 16 carbons, so it is a 16 carbon fatty acid. And now you can imagine howsoever long chain you can make, and you can make a fatty acid of that number of carbons. So 14 comes from CH2 groups, one is from CH315, and one from the carboxylic group. Okay, is it clear?
Yes, sir.
So this is palmitic acid. One more example which was asked was arachidonic acid. So if you just write these commonly asked one, arachidonic acid, and this is a 20 carbon fatty acid. So you understood that all fatty acids are a type of lipids. Yes?
Yes.
Okay. Now, have you heard of, in chemistry, have you read of bonds? Single bond, double bond, triple bond?
Yes.
Yes.
So if in a fatty acid, all the bonds are single bonds. By single bonds, I mean that the valency of carbon is saturated. For example, if there is a long chain of carbon and it's all single, and again it's all attached to hydrogen, so all these bonds are single bonds, right? Is there any scope of adding something else to this molecule? Look at this structure. These are all hydrogen attached to COH. Okay. So you see all the bonds here are saturated. Yes? Yes or no? Between the carbon, all the bonds are single. Do you think there is a scope of adding more into this molecule, this structure? Chemically? Some chemistry we are discussing now. Tell me. If all the bonds between the carbon are single, which means the structure is saturated, it has reached the maximum capacity of forming bonds because all the bonds are single. If there was, for example, a double bond here, then what would have been different? The different thing would have been this hydrogen would not have been there, and any one of the hydrogen would not have been there, right? Why? Can you explain, Iram, why?
Yeah, because the carbon has 4 valency, right?
Yes, so this carbon has 1, 2, 3, 4. All valencies occupied. This carbon has 1, 2, 3, and 4. So because of a double bond, the carbon molecule is not saturated. this is not the maximum number of bonds that it can make, because technically it has formed only 3 bonds. 1, 2, 3. 1 is a double bond. So as a bond, it will be counted 1 bond only, right? A single, like a single bond, a double bond, or a triple bond. But it is association between 2 molecules only, yes or no? So if you see this carbon, it is associated with 1 carbon here, 1 carbon here, and 1 hydrogen here. So it is associated with 3 other elements, 2 carbon and 1 hydrogen. If you break this double bond, if you remove this double bond and instead make it a single bond, then it has scope for adding 1 more hydrogen here. And this carbon also have a scope of adding 1 more hydrogen here. Now both the carbons are associated with 4 different elements. Yes or no? Do you understand?
Yes.
So fatty acids can be of 2 types. Write down. Fatty acids can be 2 types. One is called saturated fatty acids. Saturated fatty acids. You can write without any double or triple bond. And second is unsaturated. Okay. Unsaturated is with 1 or more double bond. This triple bond is not possible. So I think there's no need to write it, because in a chain, why is a triple bond not possible? Aisha, why in a chain of carbon molecule, a triple bond is not possible? Or is it possible? Can carbon form a triple bond? Yes, no, anything? So if you don't know, you have to say no. Then I will tell you.
Yes.
Yes, it can form a triple bond. So why I'm saying that in a saturated fatty acid, triple bond is not there mostly? Why? Let's try do it. If I make a triple bond here, is this possible? To make this adjustment, what I have to do?
Remove 2 hydrogen.
So I will remove this one and this one, right here. And for here also, I have to remove this hydrogen and this hydrogen, right? Yes or no? It is possible at the last carbon. So it is possible here. Now let's say, let's go a little back. Let's bring in all the hydrogen back. So to make a chain, tell me, is it possible if I induce a triple bond here? What changes do I have to make? Tell me. If I remove these 2 hydrogen and I also remove these 2 hydrogen, is it possible? Right?
Yes.
So no? Yeah. But it's very highly unlikely, because you have studied in your junior classes that a triple bond, which is the longest bond, which is the smallest bond? Are they all of the same size, same length, distance between the atoms, or are they different? What is the difference between single, double, and triple bond? No difference. The difference is of the length of the bond. Single bond is the longest bond. Double and triple. Double bond is smaller than triple bond. Smaller than single bond. And triple bond is smallest. It is smaller than both single and double bond. Is it clear? Hiram and Aisha?
Yes.
Yeah. Are you understanding? I know it's slightly boring topic, but once you get ahead of this understanding, it will be easier for you to score good, both in biochemistry and chemistry. So saturated fatty acids are those which only have single bonds. There is no double bond, which means no scope for any more addition of atoms. Unsaturated is with one or more double bonds. Now use your logic and tell me which one of the two is more stable fat. Which is more stable?
Saturated?
Saturated is more stable, right? Makes sense to you, Aisha, or do you disagree?
No, I agree.
You agree that saturated fatty acid with all single bonds are more stable. But you know, it's easier to break single bond and it's not very easy to break triple bond. Because to break a single bond, you need less energy. And to break a triple bond, you need more energy. Now think again and tell me, which of the two, saturated or unsaturated, will be more reactive? Just confusing you a little bit to get your concepts stronger. Tell me.
Saturated.
What saturated? Just don't tell me in one word. Saturated will be
more reactive.
More reactive. Then how it is more stable? More reactive molecules are less stable molecules. Now I can see that both of you are totally confused. Now is the right point, right time. Tell me, which is more stable, saturated or unsaturated?
Unsaturated.
Okay. Then why did you say saturated in the first place? The rule of stability in chemistry also says, you are forgetting chemistry. You're just, it's so easy to confuse you, both of you. The rule of chemistry also says that the elements whose valency is fulfilled are the most stable elements. Or in a compound in which the valencies are fulfilled are less reactive and more stable. Now again, I'm asking you, which one do you think is more stable, saturated or unsaturated?
Saturated.
You have changed the answer 3 times, Aisha. You better not change it the next time, okay?
No, sir. Iram said unsaturated. I did not say.
Okay, so you are saying that, but you said that saturated are more reactive. I remember that.
Yes.
So, and it cannot be, you are saying that saturated are more reactive and saturated are more stable. Does it make sense to you? It's like elephant is very big and elephant is very light. I can carry it in a pocket. So if a substance is more reactive, how it can be more stable? Reactive means it will just react with something and form something intermediate and a product, things like that. Chemistry will happen. One final call, taking everything together. Iram, which is more stable and which is more reactive? According to Aisha, saturated is both more stable and more reactive. You can change your answer, Aisha, but after Iram. Iram.
So I think saturated is stable and it's not reactive. And unsaturated, it's reactive and it's not stable.
Okay, Aisha. Aisha, your time. Aisha, you're mute if you're speaking. Which is more stable and which is more reactive?
Saturated.
She speaks in one word. What do you mean by saturated? What is saturated? I can fill in the blanks with anything. Saturated is something that Aisha does not know anything about. What do you mean by saturated? Saturated is what? More stable or more reactive? Okay, we can keep doing this all night, but let me then put an end to it. Saturated is more stable and hence less reactive. Do not react as compared to unsaturated, which is less stable and more reactive. Now I'm trying to connect it to some entrance questions now. So you understood that what is a saturated fatty acid chain and what is an unsaturated fatty acid chain. Now tell me which of the two kind of fatty acids or the fats, if you eat, if you consume, will be harmful for our body and which will be useful for our body. First you tell me. Yes, Hiram.
I think so. Saturated will be a little bad, harmful,
And
and
unsaturated is good.
yeah,
Okay. And why do you think so?
because they're stable, right?
So they are harmful. You have to so it's like I'm asking you, why is an elephant heavy? Because it's gray. Make sense? It is gray. You are right. You are correct. It's a true characterization of that elephant. Saturated are more stable, I told you. I'm asking why are they harmful if you're saying that they are harmful? What is the logic? How can you connect more stable, less reactive, less stable, more reactive with harmful or useful in our body? What kind of substances are useful in our body? So let me go through this. To make your muscles, what do you need? To grow, to make your muscles, to grow your body, to grow in size. What do you need?
Proteins.
Sorry, proteins. Yes, very good. You need proteins. But what do you eat in lunch, breakfast, and dinner? Do you eat protein? You eat a chapati or some
Yes, sir. Dal, chawal.
dal, chawal, roti, sabzi. You eat all these things. Why don't you just go to market, take protein powder, put some water in it? Because we need protein, no? Why don't we do that? Why do we eat roti? So does the roti goes and becomes our muscle? Where does roti go? This is even basic than one step at a time. The roti goes in our stomach. What happens? See, Aisha was fed up. Aisha was like, "Arey, bol do yaar, stomach." She just unmuted herself to just vent out her frustration. Did you realize the tone, Hiram? Stomach. Roti goes in stomach. She was like that. Okay. Roti goes in stomach. Good. Aisha. What happens in stomach?
Digestion.
Aisha. Digestion. What is digestion?
Breaking down of food.
Yes. So our stomach has a big hammer. So when roti comes, the stomach breaks down the. What is the meaning of that breaking down? These are some textbook lines. What do you mean by breaking down? The same way we break down bricks?
No.
No. Then what is that breaking down? How is it breaking down? What's happening inside the stomach? Oh my God. Aisha is again going to burst. Aisha, you tell. Don't get angry, okay? Tell me. What's happening in the stomach? What is present in the stomach waiting for that roti to come in?
Enzymes.
Enzymes. Very good. Aisha was like, Arey yaar, wo I said.
Sir, it's
Good.
me.
Iram. Okay, this time Iram is frustrated. Good. Mutual frustration is good for knowledge sometimes. So inside the stomach is enzymes. And what are these enzymes doing? What are enzymes?
They're breaking down.
Bak. So enzymes have this hammer and they're breaking down the roti. What is happening? What is happening? What is the logic? There is one single word that is happening. The same thing will happen if you take the same enzyme in a beaker and put a piece of roti in it. You can do the same thing in a chemistry lab. So what is happening in our stomach? God, girls, chemical reaction is happening in our stomach. The breakdown is not physical, it is chemical. You understand? Right? Our stomach and our body, every cell of our body is just a chemistry lab, nothing else. It's just a very tiny, minute chemistry lab doing chemical reactions. All these reactions, when we go in our higher studies in 11th, 12th, in our BScs, the same thing comes in our practical. We have to do that and get marks. Cells are doing it inside. So these enzymes are catalysts that break down a molecular structure. So the protein is a long polypeptide chain made up of amino acids, right? We just studied it above, right? Amino acid?
Yes.
So lots of amino acids together make a polypeptide chain. And these enzymes, for example, for protein, we have proteases in our body, in the stomach and intestine, which breaks this long chain into small amino acids. So the breaking is happening of the bonds, right? Chemical reaction is happening of the bond. So basically our metabolism is nothing but chemical reaction. Yes or no?
Yes.
Are we on the same page till here? So anything which goes in our body and can get broken down through metabolism is not harmful. We can get rid of it. Any drug which goes in our body and does not accumulates but can be broken down through metabolic pathways is not very harmful for us. If we can break down something chemically, we can get rid of it. And Iram was right saying that saturated fatty acids are harmful because they are saturated. They do not participate in reactions, right? So our body cannot break them. They do not participate in chemical reactions. Being stable, right? You understand? Iram and Aisha. Whereas unsaturated fats, if we eat unsaturated fats, they will participate in reaction. Our body, lipase is present. Lipases are enzymes which break down lipids. I'm telling you a rule here. Enzymes have the word ases in the end. So lipase is ASE. Lipase break down lipid. If I say protease, what does it chemically break down?
Proteins.
Proteins. If I say nuclease, nucleic acid, with which our DNA is made up of. Okay, nucleic acids. If I say amylase, salivary amylase, you all know. What does amylase? Yes. Okay, very good. So write down. Okay, and before I go there, also let me ask you one more question. So if we eat saturated fatty acids, they are why are they harmful? Because they are not getting metabolized. So what will happen if they will not get metabolized? What will happen? They will
increase blood.
They will not increase blood. They will increase in our body. They will keep accumulating, right? And when they will accumulate, where will they accumulate? They can accumulate anywhere, right? On an organ, in blood vessels, leading to choking of the blood vessels. Have you heard of these things that the blood vessel got choked, or the coronary artery got blocked? Some arteries gets blocked near the heart or in the heart, and then we have to do a bypass surgery. Have you heard of these terms? If you wish to become doctors, you will be reading about these terms a lot. What is a bypass surgery? Basically, one artery got choked because of accumulation of saturated fatty acids. So now you have no other option than to take another artery and make a bypass way like this. So that blood, instead of going, it cannot go like this. So blood now will go like this. This is called bypass surgery. But this is not a permanent solution or a treatment, because over time, even this can get choked, right? It is just like the concept of bypass road. Have you not heard of the term bypass road? Or flyover, expressway, bypass expressway? It's just like that. But the solution is to decrease saturated fatty acids from our body. Okay.
So,
yes,
is it related with cholesterol?
Yes, exactly. I was not using that term because I was about to use it later with the structure. But yes, cholesterol is not good for health, right? There is something called good cholesterol and bad cholesterol.
Yes.
Okay. So if it is saturated, it is bad. If it is unsaturated fatty acid, it is good. There are also terms like VLDL, LDL, and HDL. Do you know about what is LDL and HDL?
Yes.
Yes, Aisha, tell me. LDL, low density lipids. Okay. Low density lipopolysaccharide or low density lipids. HDL is high density lipid. Now, can you think which one is harmful and which one is useful?
LDL is the harmful.
LDL and HDL are two types of cholesterols. Yes. LDL is harmful.
Yes, it is a
Yes.
bad cholesterol.
Yes. LDL is bad cholesterol. And HDL is high density one is, it's called good cholesterol. So this should be in our body. Okay. So LDL is low density lipoprotein and HDL is high density lipoprotein. So lipo word comes from lipids. And you can understand why it has low density, because all of them are saturated single bonds. Yes, you understand, everyone? Both of you, Iram, Aisha?
Yes, sir.
Yes.
So write down now, keep writing certain things. Saturated fatty acids are not good for health. Whereas unsaturated fatty acids are good for health. So fatty acids, lipids can be fatty acids. Apart from fatty acids, another form of lipid is glycerol. You've read of glycerol? You know about glycerol? It is also another simple fatty acid. What do you know about glycerol?
It is an alcohol?
Yes, OL. It is an alcohol. It has alcoholic group. It is an alcohol. The structure is very simple. It has 3 carbons as a chain, and every carbon has an alcohol group attached to it. OH. So if every carbon has an alcohol group attached to it, only 2 of the valencies, in this case 3 valencies, are satisfied. So what will be the other 2? Hydrogen, right? Here it will be only 1. Here it will be 2. So you can also write this as CH2OH, CH2OH, and CH2OH, right? This is glycerol. It is another type of simple lipid. Write down. Glycerol is another type of simple lipid. And remember, if there are 3 carbon, what do we call it? What is the name given to a 3 carbon structure? Remember ethane, methane, propane, you have studied these things in
Yes.
chemistry, yes or no? So
Yes.
eth, the word eth is for ethane. Eth is 1 carbon. Meth is 1 carbon, methane. Eth is 2 carbon, ethane. Then prop is 3 carbon, propane. So another name for glycerol, which was the entrance question. The another name for glycerol is trihydroxypropane. Propane because 3 carbons are there, eth, meth, prop, and all are single, so it will be propane. If there is a double bond, then it is propene. If there is a triple bond, it is propyne. Rules of chemistry, right? I hope you know this. Please go back and
Yes,
brush up a little. Yes. So trihydroxy, because this is the hydroxyl group. OH is called hydroxyl group. There are 3 OH, so trihydroxy, and a 3 carbon chain which has single bond all through. So propane. Make sense to you? Yes. So sometimes lipids, these are types of lipids, either fatty acids or glycerol. Some lipids have both together. Lipids and fatty acid, glycerol and fatty acids together. So write down. Some lipids have both glycerol and fatty acids. Some lipids have both glycerols and fatty acids. And in this case, the fatty acids are esterified. The fatty acids are esterified. Esterification. Esters, have you studied about esters in chemistry? Ether,
No.
ester? No. Okay. Esters are sweet smelling substances. But just write down, in this case, when in a lipid, a glycerol and a fatty acid are both present together, so this glycerol component will esterify the fatty acid. So what have you written? The fatty acids are esterified with glycerol. So if both glycerol and fatty acid is present together, then we call it either fats or oils. You know what is the difference between fats and oils? You both know, but I don't know if you will tell it or not. What is a fat and what is a oil? In both fats and oils, glycerol and fatty acids both are present together. And in both of these, it is esterified. The fatty acid is esterified by the glycerol. But what is the difference between fats and oil? Both of you, I assume, eat non-veg?
Yeah.
Yes, very good. There's not much in chicken. Let's say mutton. Let's take an example of mutton korma. Cool? Tasty example. Yes? So I'm giving you the hint that in mutton korma, both fats and oils are there. Yes, Aisha. Now bring back your neurons to focus on some concepts. Hero has said hard only. Okay. I believe you love food. So now can you tell me which component of a mutton korma is fat and which is oil? Yes. Tell me. Have you seen raw meat? You have
Yes.
cleaned, washed raw meat. So there's this yellow, slimy thing present with the flesh, with the protein part. That's called fat. Fat of the animal, right?
Yes.
Yes.
When humans get obese, they have fat deposition in their body. That's fat. So that fat is present in the meat. And you cook the meat in oil. So the basic difference between fats and oil is that fats are solidified. You understand? And oils are liquefied. So the mustard oil or the soya oil, these are oils. But if you see ghee, or if you see butter or cheese, they are fats. You understand the difference, Aisha and Iram?
Yes, sir.
So oils have a lower melting point. That's why they are liquid at room temperature. Lower melting point. And fats have higher melting point. They also become liquefied, but at high temperature. Higher melting point. And again, when they come to low temperature, they again solidify. Right? Best example is butter. If you keep it in refrigerator, it solidifies. But if you take it out and keep it on the pan, it liquefies. Yes or no? But oil stays liquid. It has a lower melting point. Is it clear?
Yes, sir.
Yes. So write down. So this is the third kind. And in both, in fats and oil, you can just write down. Both glycerol plus fatty acids are there. Okay. And fats and oils are also types of lipids only. So fat, oil, glycerol, fatty acid, all are types of lipids. Do you understand the concept now? Are you clear with this, Iram and Aisha?
Yes.
Palmitic acid is also a fatty acid, hence it is also a lipid. Arachidonic acid is also a fatty acid, hence it is also a lipid. So if I ask you, all fatty acids are lipids, but all lipids are not fatty acids, is this statement true?
Yes.
Yes.
So you just solved a neat question. Very good. So you understood all fatty acids are lipids, but all lipids are not fatty acids. Lipids can be glycerol, lipids can be fats and oils, etc. Write down some lipids, the next type. Some lipids very good. Yes, Iram is celebrating that she solved the question. Good. So it's all just the concepts, nothing else. So some lipids, write down, contains phosphorus. Okay. Contains phosphorus or a phosphorylated compound, organic compound. Phosphorylated organic compound means any compound that contains phosphorus. Simple. So either they contain directly phosphorus or a phosphorylated organic compound. And in this case, these lipids will be called, you have heard the name, phospholipids. Right? These are called phospholipids. You understand? These are not oils, these are not fats, these are not glycerol. These are phospholipids, lipids that contains phosphorus. Okay. And they are the ones that we study in the cell membrane. Phospholipids are present in cell membrane. Do you understand now what our cell membranes are made up of? They are made up of a special kind of lipids known as phospholipids. That fluid mosaic model that we studied. Oils, the examples of oils. Also, let's keep writing some examples. Oils are, can you give me some example of oils? Mustard oil, gingerly oil. Okay, fats, butter, ghee. Phospholipids present in the cell membrane. And the example is lecithin. Lecithin is one phospholipid. Hence, lecithin is also a lipid. Lecithin. Different types of lipids are clear to you?
Yes, sir.
Okay. There are more complex types of lipid than this, but I think that we do not have to study in detail. So just remember phospholipids, fats, oils, glycerol, and fatty acids. Okay, everyone? Both of you? Fine? So lipids is over. Now next, what is the next biomolecule? What should we go to? Let's go to nitrogenous bases. They are also biomolecules.
Nucleic acid?
Yes, nucleic acid is made up of nitrogenous bases. We are going to the basics, molecular level. When we studied protein, we did not study protein. What did we study?
Amino acids.
Amino acids, right? Yes. Proteins are made up of amino acids. So proteins as a biomolecule are made up of amino acids. When I studied lipids, we studied not just the lipid, but also what are the different parts, right? So then we went to lipids, and lipids can either be fatty acids, okay, or glycerol, or combination of both, which are fats and oils, or phospholipids, which are present in plasma membrane. Now we are reading learning what makes nucleic acids, okay, which I have written as a biomolecule here at the top. If you see nucleic acid, so they are made up of bases, right? Nitrogenous bases. Now what are how many types of nitrogenous bases are there? How many nitrogenous bases are there? We have studied DNA, right? Have we or not? Okay. There are 5 types of nitrogenous bases. Adenine, which we write as A, right? ATGC. Remember this? ATGC? Yes or no?
Yes.
And guanine, adenine, guanine, cytosine, thymine, and the last one is uracil. Now, out of these 5, the first 4, ATGC, adenine, guanine, cytosine, thymine, is present in DNA. Okay, this thymine is present in DNA only. Or let me Don't write this. I have a better way to write this. So the first three, adenine, guanine, and cytosine, can be present in both DNA and RNA. Is it clear? This is common. A, G, C is common. But we need 4 to make pairing. So thymine is only present in DNA. And it makes bond with A. And uracil is only present in RNA. And again, uracil forms bond with adenine. Is it clear? The 5 types of bases, nitrogenous bases. AGC is present in both DNA and RNA. Thymine only in DNA, uracil only in RNA. Okay. Clear? Move forward. Now, these nitrogenous bases are cyclic, heterocyclic ring compounds. What does the word hetero means? Whenever in biology we are confused, we go to the word, the terminology. What does the word hetero means? Different, right? Heterogeneous mixture, homogeneous mixture. Am I making sense, Iram?
Yes.
Heterocyclic ring structure. And let me show you what is the ring structure like. You have NCERT, both of you? You can go to NCERT and see that there will be some structures given. Let me insert this diagram. Yes. Can you see this diagram? Is this clear? The diagram is clear to you? See, this is adenine. Okay. And adenine and guanine, A and G, these two are called purines. They have similar structure. A and G, adenine, write down. Adenine and guanine are called purines. Purines have a dicyclic structure. See, they have given an example of adenine here. Look at this structure. Can you see in this cyclic ring, there is nitrogen, and where they have written nothing, it's obvious that there will be carbon, right? So at these places is the carbon, and on other places is nitrogen. So there are 2 different kind of compounds making the ring. So that's why it is called heterocyclic ring structure. If a ring is made up of only carbon, it is not heterocyclic. You understand? Iram, Aisha? We call them just cyclic. For example, a benzene ring. You have studied benzene in your chemistry?
Yes.
Yes.
What is benzene? A 6 carbon ring, right?
Yes.
Which has alternate double bonds like this. They are not fixed. So this is called a cyclic compound. All are carbon. But a heterocyclic has more than one type of compound making a ring. So see, look here. So adenine and guanine both are purines, and purines have this kind of structure. 2 rings are there. One is a 6-membered ring, another is 5-membered ring. So this is 6-membered ring and this is 5-membered. Whereas, write down, cytosine and thymine. Cytosine, thymine, and uracil, all these 3. Cytosine, thymine, and uracil are pyrimidines. They are pyrimidines. So this is the structure of uracil here. They're pyrimidines. The difference between thymine and uracil is if you just put here a CH3, it will become thymine. If this CH3 is not here, it is uracil. But there is only one single ring, unicyclic. This is dicyclic, this is unicyclic, pyrimidines. So these are nitrogenous bases. These are bases. Now, is this clear? The structure of bases, nitrogenous bases?
Yes, sir.
Now, when these bases are present with sugar, okay, so this compound is present with a sugar molecule, then it's called nucleoside. So write down nitrogenous bases when nitrogenous bases if attached to a sugar, nitrogenous bases if are attached to a sugar, then they are called nucleosides. They are no longer called nitrogenous bases now, they are called nucleosides. So look at this here, this thing. So they have not made the structure, but they have written uracil here. So you know that this uracil is basically this thing, right? And here they have written adenine here. So it is this structure. So this structure, imagine this structure, it would be very complex if they would have drawn it. So they have just written adenine. And can you see this 5-membered ring here? 1, 2, 3. So there are 5 carbons. 1, 2, 3, 4, and this is the 5th one here. 5 carbon, 1 oxygen. So this sugar is called pentose sugar. The word pent means 5 carbon. Here also it's the same, 1 carbon, 2 carbon, 3 carbon, 4 carbon, and 5 carbon. So these things are not given in NCERT, but they are asked in entrance. So this is a pentose sugar, and the name of the sugar is, you will get to know the name of the sugar if you will tell me the name of the full form of DNA and RNA. What does DNA stands for?
Deoxyribonucleic acid.
Deoxyribonucleic acid. So DNA. What is RNA?
Ribonucleic acid.
Ribonucleic acid. Perfect. Very good. RNA. The first part tells you about the sugar. In DNA, the name of the sugar, the pentose sugar, is deoxyribose. And in RNA, the name of the sugar is ribose. So sugar is deoxyribose. And in this case, the sugar is ribose. Because apart from the sugar, other thing is nucleic acid. Okay. So you understood, both deoxyribose and ribose are pentose sugars. They have the same kind of structure like this. So when it become a nucleoside, their name changes. Adenine become adenosine. And uracil become uridine. Adenosine and uridine. What was the full form of ATP, which mitochondria used to make in a cell? I'm connecting knowledges here. What is ATP?
Adenosine triphosphate.
Sorry, come again, Aisha. Be a little clear. Hiram, Raisha, I'm not able to hear Raisha.
Phosphate?
Yes, adenosine triphosphate, right? And when I explained this to you, I made this one gola, wrote A in it, and said there are 3 phosphate groups, right? Now you know what is this A, what is this adenosine? It is this thing, right? Adenine plus a pentose sugar is adenosine, then 3 phosphates. You understand? So the adenosine is a nucleoside. Clear what is nitrogenous base and what is nucleoside? A nitrogenous base plus a sugar is nucleoside. And in our DNA and RNA, it is a pentose sugar. In DNA, it is deoxyribose, and in RNA, it is ribose. Clear till here?
Yes, sir.
Then there's third thing called nucleotide. Write down. A nucleoside, when attached to, so what is the difference? You tell me. There is a adenine which is attached to a 1, 2, 3, 4, 5 carbon sugar. So adenine is there. Pentose sugar is there. What is additional? Can you see this phosphate group? This group? This is called phosphate group. This is additional. So a nucleotide, oh sorry, write down. A nucleoside, when attached to a phosphate group, a nucleoside, when attached to a phosphate group, becomes a nucleotide. Becomes a nucleotide. Is it clear? Iram and Aisha? So in a flow chart, let me write you like this. So in the beginning, we have nitrogenous bases. If you add sugar to the nitrogenous bases, then these two together will be called a nucleoside. Okay? And if you add a phosphate group to a nucleoside, then this together will be called a nucleotide. Is it clear? This thing here. Yes? And this nucleotide is the, write down, it is the building block. Building block of DNA and RNA. It is the monomer. You can also use the word monomer, because DNA and RNA is a polymer. It's a long chain. Just like protein is a polymer of amino acids, you understand? Hundreds and thousands of amino acids are linked one after the other to make a protein chain. So protein chain is a polymer of amino acids. Similarly, a DNA or RNA is a polymer of nucleotides. Okay? So DNA and RNA is made up of nucleotide only. So you understood what is a nitrogenous base, what is a nucleoside, and what is a nucleotide. Can you differentiate between all three now? Yes, you know, very good. And we know that DNA and RNA, just add one more line. DNA and RNA is the genetic material in a living system. DNA and RNA is the genetic material in a living system. Okay. Now, all of everything that we have studied till now, what have we studied till now? We have studied proteins like amino acids, lipids, nucleic acids, sugars also we have studied, right? Sugars are, if I show you the structure of sugars, let me see if I can show the structure of sugars. Sugars are basically carbohydrates, right? So let me also come to, so last part, the last molecule that we have to study is sugars. I call it sugars, or I call it carbohydrates. All sugars are carbohydrates only. Now, let me go to another image. Please open your NCERT, the same image. There will be something about sugars also. Yes. I don't need the full figure. Just one. Give me 1 second, Hiram. Let me pull out that figure for you. Yes. Can you see this image that I have put in here? It's from your NCERT only.
Yes.
So sugars, I just told you about ribose, right? 1 carbon, 2, 3, 4, and this one is the 5th one. So ribose is C5H10O5. Can you see the formula here? And because there are 5 carbons, it is called a pentose sugar. Another kind of sugar is glucose, the most common energy source in our body. Yes. Which are the only type of cells which do not use glucose as energy in our body? I have this bad habit of asking questions from previous chapters. Tell me. Gamete cells, sperm, remember? Sperm use fructose as energy source, not glucose. Iram. We discussed this, right? When we were studying cell. Yes or no?
Oh.
No. No?
Yes, no, we didn't.
Okay. I'll check. Okay. Then just know that in human body, there is only one type of cells, which is sperms, the male gametes, that do not use glucose as an energy source, but use fructose as an energy source. Apart from those, all the cells of our body use glucose as an energy source. The formula is C6H12O6. So how many carbons are there in glucose sugar? It is also a ring. You can see 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, and the 6th carbon. So it has 6 carbons. So what will be the name of this sugar? If 5 carbon is pentose, 6 carbon will be?
Hexose?
Yes, see, you already know it. I didn't tell you. You just know that glucose is a hexose sugar because it has 6 carbon. So is sugar clear to you? It is a cyclic ring of carbon with oxygen. Depending on how many carbons are there, we name the sugar as pentose, hexose. That. These are the most common one. Pentose and hexose both. Glucose and ribose is present in our body. Ribose is used in the making of nucleotide. Remember, it is a component of nucleotide. Also nucleoside and nucleotide both, right? Yes or no? Look here. This is that sugar that we are talking about. It's present in nucleoside and nucleotide both. So sugars is clear. You can write down sugars are carbohydrates. Write down sugars or carbohydrates are instant source of energy during metabolism. Are instant source of energy during metabolism. That's why whenever someone is low on energy or someone got unconscious or was feeling weak, what do we give that person? What do we make that person eat or drink? Chicken? Dal chawal?
No.
Yes, what will you do then? If the person can drink, you will make him or her drink a Glucon-D, a glucose drink, right? Anything that contains glucose. Make him eat Parle-G biscuits or any basic biscuits that contain glucose. You understand?
Yes.
If the person is unconscious, we will put drips in the veins, intravenous drips of glucose, right?
Yes.
Because glucose is instant source of energy. It just goes and starts oxidizing in the cell. That is called the process of breaking down of glucose, taking a hammer and breaking down glucose. No. In the presence of oxygen is called the process of breaking down glucose chemically in the presence of oxygen is called what? It's called respiration. That happens in each and every cell of our body to produce energy. So it is the instant source of energy. So proteins, building blocks, lipids, stored form of energy, fat. They have very, very high calorific value, but they are not immediately used. So if a person is eating good food, eating enough carbohydrates in a day, plus on top of it is also very, very, fond of fatty things, fat-based things, then the carbs will get used instantly, which will be enough for you to survive through the day. And the fat, if not required, then it will be deposited. That's where obesity comes in. So people who eat more, who get more nutrition than they require, so they only burn the carb part of it. The fat gets deposited. Then to burn the fat, they have to run or do vigorous exercises or do all sorts of things to lose weight. So first we eat fat, then we don't use it, we deposit it, then we become obese, then we try to burn it. Easier way is don't eat it in the first place if you are not so active. Okay, don't eat it more than it is required, because we humans are very, very I don't know, sometimes what we do biologically is funny. We are taking more than required, then it is getting, so body, what will body do? Body is not going to get rid of the fat, right? Through fat is a important thing, right? You need it. It is not a harmful substance. So body will say, okay, I got fat, so but I also have carbohydrates. So this person ate fries, but the fries were deep fried. So it is potato basically, which is carbs, but it is fried in a oil, so it also has lipids. Plus on the top of it, there is, he or she drank cola and had a burger with a cheese, sometimes a double cheese. So you got everything, and it was a chicken burger. So you got protein, you get lots and lots of carbs, you got sugar from the cola, and you also got lots of lipids. Now proteins will go to the cell to synthesize, repair, wear and tear, to make muscles, etc. The carbohydrate component will go to the cells again as glucose or ribose and will be used in the cell to generate energy. but the fat component, the lipid component is not required. You don't need to burn, because you had that, you came back home and you slept. You're not active. So you start becoming obese over time. And then when you are um, when you are tired of the same thing that you only stored in your body, then you want to become active. So instead of that, have a restricted, contained diet. Don't eat more than you need, because then you will struggle to get it out of your system. It's very simple logic. Okay. And fat metabolism is done under conditions where you need more energy. Fat is not burned under normal conditions. So if you are walking, have you seen people, again, one more behavior. So after the evening dinner, I have seen lot of people walking. They just keep walking for kilometers, and they're thinking that they're burning their fat. Fat metabolism does not begin unless and until you are very high on activity, unless and until your heartbeat crosses 120 or 140 beats per minute, which means you are running, or you are playing a sport, or you are jumping on a rope or something like that. If you are just walking, and talking, enjoying, you're not burning fat. You can keep walking for 4 days continuously. It will not burn your fat. It will just make you tired and burn carbohydrates. You will feel tired. You will feel again hungry. You will go back, and instead of eating carbs, you will eat fat again. So that's not helpful. So we actually get in a loop. That's why getting out of obesity is very difficult. Getting in is very easy. Okay. So right from the beginning, or whenever anyone wants to start doing it, they have to be more logical with that regime. So everything is clear. The sugar component is also done, right? Now write down everything here. I will appreciate if you draw this, whatever is on the board. Just let me make it a little big here. Yes. Please draw this. Note everything down. I'm giving you 4 minutes to revise amino acids, lipids, nitrogenous bases or nucleic acid, nucleosides and nucleotides, and sugars. And then I will start with the next part, because whatever we studied till now are called primary metabolites. The primary, all these 4 things: sugars, nucleosides or nitrogenous bases, amino acids, and lipids. They are primary metabolites. Okay. So just take your time, take 3-4 minutes and revise everything. And let me know if I need to scroll up or down, if you want to note something. Both of you have noted everything down, which is on the board?
Yes.
Yeah. Okay. Now quickly revise. Okay. I'll be back. Okay. Iram, Aisha, cool. Are you revising?
Yes, sir.
Okay. Okay, are you there? Both of you? Done revising? Everything is clear? Because if anything is not clear, please ask. If everything is clear, I'm going to make it a little more complex because we have just started with the basics here. Everything is clear, Iram and Aisha?
Yes, sir.
Yes? Okay. Now, when we talk about biomolecules, whatever we have studied till now are the most basic ones: amino acids, sugars, lipids, and nucleotides or nitrogenous bases. Now, in a animal tissue or in a plant tissue, all these compounds that we have studied till now, including cholesterol, which is HDL, LDL, glycerol, fatty acid, amino acids, nucleotide, nucleoside, all these things, they are called primary metabolites. As you go, so the primary metabolites are the ones which are present in all cells. Okay, these have to be present. If something is primary, it has to be present there. Then comes secondary. Secondary things are like a little advanced, little complex, but they are present only when they are required. Like they are not required for survival. You understand? Like the primary thing is that everyone should have a primary education, like a basic education where they can read, write, understand. Secondary or higher education depends on what are you interested in. So an engineer will not study about genetics or cell metabolism, etc. And a biologist will not study about astronomy and things, because only the things which are required you will. But both a cosmologist, a biologist, an engineer, a doctor, what is common among all of them? Their primary education, the school level education. Yes or no? Everyone has to go through the same schooling. Yes or no? Especially the primary schooling. So till 10th, whoever you are, whatever you become in life, till 10th, you study the same things, right? Aisha and Ira?
Yeah.
Yes, sir.
Yeah. And after 10th, you are equipped to be able to read, write, and understand anything given to you. Yes or no? So you are educated to some extent. Now for secondary and higher education, you can choose whatever you want to go into. So similarly, write down primary metabolites. Primary metabolites are those biomolecules which are present in all tissues. In all tissues. Okay. Especially the animal tissues. They are present in all animal tissues. So primary metabolites are those biomolecules which are present in all tissues. Now we are coming to primary versus secondary metabolites. Secondary metabolites, on the other hand, write down, there are many compounds other than there are many compounds other than the primary metabolites which are known, which are present in plant, fungi, and microbial systems, which are present in plant, fungi, and microbial systems. Okay, so they are present mostly outside the animal system in plant, fungi, and microbial systems, and are known as secondary metabolites. And are known as secondary metabolites. Next point, I'm just making you write the difference between primary and secondary metabolites here. I hope you are understanding, right? Iram, Aisha?
Yes, sir.
Write down. Examples of secondary metabolites are. Examples of secondary metabolites are. Antibiotics, pigments, scents, spices, rubber, resin. Scent, spices, rubber, resin. Alkaloids, 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 essential oils, etc. Essential oils, etc. Okay. Now, one another difference is that for the primary metabolites, we know that they are so crucial, so important, that we know their roles and their functions in normal physiological process. But we do not understand completely all the functions of the secondary metabolites in the host organisms. Like they are useful to us, like we take it out and use them. For example, we make rubber from the like the latex, right? Tree secretes this rubber, the latex material, which we collect and convert it into rubber. Yes, you understand? Aisha, Iram, where do we get rubber from? The natural source of rubber is the tree which secretes this resin, the rubber tree, right? So we use it for our welfare, but we do not know exactly what are all the roles of that resin in the plant system. What is the plant system doing with it? Similarly, let's see here. So these are examples of some secondary metabolites. So these could be pigments, carotenoids. Carotenoids are the pigments which are present in carrots. So it is a pigment, colorful pigment, anthocyanins, but we do not know. We know the functions of some, but not of all. Not of all of them. Then secondary metabolites could be alkaloids like morphine, codeine. We know that it acts as a drug, right? You have heard of morphine? Morphine acts as a painkiller.
It is used after surgery.
Sorry, come again.
It is used after surgeries, the morphine.
Yes. Earlier, morphines were. Now we have better analgesics, aspirin, etc., ibuprofen. So and to give the anesthesia, we use better substances than morphine, because morphine is also addictive. Codeine, morphine, these are addictive substances. They act as hallucinogens and painkillers, right? But what is a plant? Plants that produce it, what are they doing with this substance? They are not protein. They are not important substances like protein or lipids. which help in the making of the cell membrane or the protein, like the structural things in the cell, or they are not nucleic acids which make the DNA, but they are still there. So they are called secondary metabolites. Third is terpenoids, terpene oils, monoterpene diterpenes. They are called terpene oils. You know where is terpene oils used?
Paints.
Yes. So we use terpene oils as a thinner of paints. We mix it with paints and it dilutes the paints, right? But why is that system producing it? Did it knew that one day humans will have to paint their homes? So let me, no, right? Let me start producing terpenes so that one day they can paint their homes. No, right? We are using it for our benefit, but their biological relevance is 100% not understood in all the cases. Fourth is essential oils like lemongrass oil. Also toxins, abrin and ricin. They are toxic substances present, produced by systems. Then lectins are examples. Concanavalin A is a lectin. Certain drugs like vinblastine, curcumin, they have human uses. We use all these things. Polymeric substances like I told you, rubber, gum, cellulose. We know that for cellulose, we know that plant cell wall is made up of cellulose, right?
Yes.
So this we know that why they produce cellulose. And we have also figured out use for others. Why, like toxins, why do systems produce toxin? To protect themselves. But for every secondary metabolite, all the functions, physiological functions, are not known completely. Is it clear? So write down the primary metabolites have well understood functions and roles in normal physiological process. Primary metabolites have well understood functions and roles in normal physiological processes. Whereas the functions of all the secondary metabolites in the host organism means where it is produced, their function for us is known. Why do we use rubber? To make rubber bands or to make all the things which are made up of rubber. But for the plant I'm talking. So the function for all the secondary metabolites in the host organism is not properly understood. Okay. And last point, secondary metabolites are very useful to human welfare. Secondary metabolites are very useful to human welfare and also have ecological importance. Is it clear, Aisha, Neeram?
Yes.
Okay. So we have done the the first level of the chapter, where we studied about primary metabolites in details and secondary metabolites in not so much of detail because it's not in your syllabus. Now, from the next class, we will start building on it. So what did we study? Amino acid, sugars, nitrogenous bases, nucleotide, nucleosides, and your lipids. Now we will go up and study about biomacromolecules. All amino acids make what?
Proteins.
So we'll study about proteins. All the sugars make what?
Carbohydrate.
Which is called polysaccharide. So we'll study about protein, polysaccharide, fat metabolism, and nucleic acid, and then their digestion, their metabolism in the body. Okay. So this part, if you understood the basic now, the next part is going to be like not very easy, but you will connect to it with much ease. And then in the end,